Hello, ladies and gentlemen. Mrs. Grider here to talk about linked genes. What happens when genes are located on the same chromosome? So, to start off, remember what we've been talking about for the past few days. We've been talking about independent assortment in meiosis. So, independent assortment is when chromosomes line up randomly during metaphase 1. So, we can see here in this example that there's two potential arrangements for these chromosomes. It's independent assortment because how these homologous chromosomes on the top line up has no influence on how the homologous chromosomes on the bottom line up. And the end result of this is that we get different combinations of chromosomes in the gametes. In this example, 4 different combinations of chromosomes. This has a big influence because if we have genes located on this pair of homologous chromosomes and genes located on this pair of homologous chromosomes, we're getting new combinations of those genes in each one of the gametes here at the bottom. But what if, instead of being located on different chromosomes, these genes were combined onto the same chromosome? That's what we're going to be investigating today. So, in order to do this, we're going to talk about our friend, the humble fruit fly. Fruit flies have been used for over a century as what's called a model organism. A model organism is a common test subject for experiments that lots of scientists use, and then we extrapolate what we find from that model organism to other organisms that exist in the world. So, fruit flies are great model organisms because they're small, they're cheap to take care of, they have a really short life cycle, so from the time they're born to the time they start breeding is really fast. They have lots of offspring, few chromosomes, which is helpful in genetics because it limits how many options there are. And now at this point, we know a lot about fruit flies. So scientists that work on them have a wealth of information that they can use in their own studies and then continue to study from. In order to talk about fruit flies, we have to talk about a new system of denoting our genes. We've been talking about Mendel's system previously, and Mendel uses uppercase and lowercase letters to denote which genes are dominant and which genes are recessive. The letter is determined by the dominant trait that we're studying. So an example that we've talked about is capital T is tall, lowercase t is short, the tall gene is dominant, the short gene is recessive, and then we can get these combinations of genotypes as a result. Today we're going to be talking about one of the first prominent fruit fly experimenters. His name's Morgan, and he has a different system for denoting the genes. He wasn't a huge fan of Mendel. He was quite skeptical at the beginning, so he invented his own system. So in Morgan's system, all of the letters that we see are going to be lowercase. The letter is going to be from the mutant phenotype, and Morgan uses a plus to show the wild type or normal phenotype. So we're going to be looking at wild types versus mutants instead of dominant versus recessive. Morgan's system doesn't actually show us whether something is dominant or recessive. We're going to have to kind of logic that out from what we see in the results of the crosses. So, an example of Morgan's system that we'll talk about today is vestigial wings versus normal wings. Here's two pictures of a fruit fly. Here's a normal fruit fly down here. Normal is also known as wild type because it's what we would find in the wild if we caught some fruit flies. So a wild type genotype has this long wing that allows the fruit fly to fly and get around. The mutant phenotype has these tiny wings. They're called vestigial wings, which means they're short, they're useless, they're there, but they're not functional for the organism. So the letters that we're going to use to track wing genes will be VG because vestigial is the mutant phenotype, and that's how Morgan determined which letters to use, based on the mutant rather than the wild type. So here we can see the letters will be VG, and if we add a plus sign, that will be for normal wings. Without the plus sign, that will show vestigial wings. The potential genotypes we can have then are 2 normal wild type alleles, a heterozygous wild type and mutant allele, or 2 mutant alleles. All right, so if we're looking at this first cross that we're going to investigate in the P generation here at the top, who's wild type in the P generation? Well, I'm going to look for the plus signs. So I see that over here on this side, I've got B's with plus signs and VG's with plus signs. So that means that this fly on the left is the wild type fly because it's denoted with the plus signs. Over here on the right, I see a genotype with no plus signs, so they must be the mutant. So the traits that we're going to be looking at today, the wild type is a gray body with normal wings. The mutant is black with vestigial wings. In this case, this is a double mutant, so both traits are mutant traits. This is a double wild type, so both traits are wild type traits. They're going to have some offspring, the dihybrid that we see right here. So in order to figure out which traits are dominant, we're going to have to look at this dihybrid wild type. It's a dihybrid because it's heterozygous for two traits. I can guarantee that it's heterozygous because I crossed a homozygous wild type with a homozygous mutant. And because we get 50% of genes from one parent and 50% of genes from the other, that means that they're going to get one with a plus sign, the wild type, one without a plus sign, the mutant. So, because they're heterozygous from this, I should be able to tell which trait is dominant and which trait is recessive. So I can see that with a heterozygous genotype, B plus B, that this fly got a gray body. That must mean that the gray body is dominant over the black body. Additionally, with this heterozygous genotype for wings, it must mean that normal wings are dominant because normal wings showed up when we crossed the normal and the vestigial. All right, once we have that dihybrid wild type individual, we're going to be doing, the dihybrid wild type individual down here, we're going to do a test cross with it. A test cross is when we breed an individual with a known homozygous recessive individual. And it's going to be homozygous recessive for all the traits that we're testing. You'll notice that the double mutant is our homozygous recessive individual. And that makes sense because they have two recessive traits, which must mean that they've got the heterozygous genotype for both of them. So our test cross is shown here in this yellow box. Our dihybrid F1 wild type with the double mutant over here. Let's see what we're going to get. So the thing that we're going to try to do first is to make some predictions in the test cross. We're going to predict the offspring from our dihybrid with our mutant using meiosis and a Punnett square. So thinking about this dihybrid fly and the law of independent assortment that we know about, we want to think about what would the two possible arrangements of the tetrads be for meiosis of the dihybrid fly. Test yourself and see if you can figure it out. Here it is. So for the dihybrid fly, we know that we've got two genes. And if we're assuming that the law of independent assortment is happening, these genes would be on separate homologous chromosomes. So up here, we have our wing gene, and down here, we have our color gene. And there's two potential arrangements of those chromosomes. You can see here that I've got the wild types on one side and the mutants on the other. Over here, I've got a mismatch of wild type on one side with a wild type on the other, a mutant on one side, a mutant on the other. Two potential arrangements of these chromosomes during metaphase 1. So pause, test yourself, see if you can figure out what the rest of meiosis would look like for each of those setups. Here it is. I'm looking at the full meiosis picture for what would happen in these two arrangements of chromosomes at the metaphase plate. So I can see that I've got a variety of gametes here at the results. Here's my 4 possible gametes. I can have 2 wild type traits, I can have 1 wild type and one mutant trait, or I can have 2 mutant alleles. 4 different possibilities that could happen. If I look at the homozygous recessive fly, I'm only going to see 1 type of gamete because it's homozygous for all of the genes that are involved. So because I don't have a variety of alleles, it really doesn't matter how these things line up on the metaphase plate. No matter what happens, I'm going to end up with these 4 gametes here at the bottom, which all have identical alleles inside of them. A side note about Mendel's laws of meiosis that we've been talking about. So we talked about that Mendel has 2 laws, the law of segregation and the law of independent assortment. Figure out right now which one still applies to what we're looking at. If you said the law of segregation, you're correct. The law of independent assortment doesn't matter here because there's only one way for the chromosomes to line up. So even if these chromosomes flip from side to side, it's going to have no bearing on the gametes here at the end. But the law of segregation is still at play here. The law of segregation says that the alleles on the chromosomes have to separate during meiosis so that we end up with haploid cells here at the bottom. And we can see that that still happens. The homologous chromosomes separate, the gametes that result are haploid instead of diploid. So segregation is still happening here at the metaphase plate, even if the law of independent assortment has no bearing on our final results. Okay, back to figuring out the results of our test cross. So here's one of those Punnett squares for this test cross, and you'll notice that because we had 4 potential gametes, it's got to have 4 columns right here. Here's my dihybrid parent gametes. I like to list them from most dominant to least dominant, helps organize my brain. And here are the recessive parent gametes over here. All of them were the same, so they're identical as we go down. See if you can figure out what the results of this Punnett square would be based on what we've done in class. If this is your Punnett square, then you are correct. And if we look at the results of this Punnett square, we see that everything in this column is identical. These 4 potential genotypes are identical, these 4 are identical, these 4, and these 4. So our results show that 4 out of 16 would have normal wings and a gray body, also known as a double wild type. 4 out of 16 a normal wings and a black body, 1 wild type and 1 mutant trait. 4 out of 16 vestigial wings and a gray body, 1 mutant and 1 wild type trait. And 4 out of the 16 have vestigial wings and a black body, which is a double mutant set of traits. The ratio of offspring that we would expect to see is that we have 4 out of 16 for each of those, which is a 25% chance if I convert that to a percentage. So the ratio is 4 to 4 to 4 to 4 for each of our potential phenotypes. Researchers often simplify this to a 1 to 1 to 1 to 1 ratio because it's identical. We expect to see equal numbers of each of those phenotypes. What does this mean for the offspring? That means that the percentage of each we would expect to be equal. We would have 25% of each potential phenotype, approximately equal numbers of each phenotype. So that means if we had 1,000 baby fruit flies, we would expect to see an equal number, 250, of each of the potential phenotypes. So Morgan does this experiment. He crosses them, he sees what happens. And here's what was observed. Here's our test cross in the yellow box. Here's the 4 potential phenotypes, and we see that we do not have equal numbers. In fact, Morgan notices something really odd. He sees that we have a lot of double wild types, 2 wild type traits, normal wings, gray bodies. We have a lot of double mutants, black body and vestigial wings, and we have very few of 1 wild type and 1 mutant trait. You can see that right here, 965 and 944 of the double wild types and double mutants, and over here, around 200 of 1 mutant and 1 wild type. So what is going on? Why is independent assortment not working like we would have expected? So here's a good place to take notes. If both genes were on the same chromosome, this means that these genes would what we call linked together. This is what it looks like here in the green star. These genes are located on the same chromosome instead of being on separate ones like we initially predicted at the beginning. This is going to mean that meiosis will look differently. If the genes are linked, then meiosis is not really looking at 4 chromosomes anymore for these traits. We're looking at just 2 chromosomes for the 2 genes. We see that they'll split up, law of segregation still applies, and we'll get 4 gametes here at the bottom. But now we're just looking at 2 types of gametes. Gametes that have both of the wild type alleles and gametes that have both of the recessive alleles. You might be wondering how I knew from the start that the wild types would be on the same and the mutants would be on the same. It goes all the way back to our parents up here. Our parents here had both wild type traits and both mutant traits here in the parent generation, so I can predict that they're going to be together from the start. So here's meiosis, 2 types of gametes. That actually changes what we would expect to see in the offspring of our test cross. So the new Punnett square for our test cross shows 2 types of gametes for the dihybrid parents, and once again, our recessive parent gametes for the double mutant. If we fill that out, we get a really different result. Instead, we get 50% of our flies expected to have normal wings and a gray body, 2 vestigial traits, or 50% of our flies with vestigial wings and a black body, 2 mutant traits. And actually, this almost accounts for what Morgan saw. A vast majority of his either had 2 wild type or 2 mutant traits. Here are the genotypes that it accounts for. But what about the ones that it didn't account for? What about these ones that ended up with 1 mutant and 1 wild type trait? This is because of the effect of crossing over. In this, we're seeing what we call recombinant chromosomes. A reminder, crossing over happens when homologous chromosomes line up in metaphase 1, and they actually physically switch part of the chromosomes. So here in this picture, you can see that these parts of the chromosomes were swapped from one to the other, and we get these inner chromatids that have some of the original chromosome and some of the other chromosome. This means that in the gametes that we'll see, we'll actually start to see 4 types of gametes again. We'll see ones that have 2 wild type traits, 2 mutant traits, and then these what we call recombinant chromosomes in some of the gametes that experienced crossing over. Why don't we get an equal number of each type of gamete when crossing over happens? Well, that's because crossing over is totally random, both if it happens at all and where it happens on the chromosome. So it's more if it happens here at the bottom with these genes, then yeah, we'll see crossing over happen and we'll get these recombinant chromosomes. But if it happened here on the other half of the chromosome, above the centromere instead of here below it, we actually wouldn't see any effect at all in these genes. Scientists like to look at this and we talk about the strength of a linkage. Is it strongly linked or weakly linked together? And linkage is determined by how close together this is on the chromosome. For example, if we look at here, gene 2 is likely to be inherited with gene 3 because they're really close together. Crossing over is just less likely to happen directly in between those 2 genes. However, we compare that to gene 4 and gene 3, they're less likely to be inherited together because they're located farther apart. It's more likely that crossing over could happen within this larger space than within the smaller space above it. So here's what actually happens during this cross that Morgan sees. Meiosis happens and we get some parent chromosomes that have no crossing over and appear just like we would expect them to. We have some that experience crossing over, but that's random, so it doesn't happen as often as the parent genes just sticking together. In the end, we see then a majority of parent-type offspring that have no crossing over that get the linked genes together, and then we have a minority of recombinant offspring that have the chromosomes that have been through crossing over, but less of them because it's random whether it happens or not, and it's random whether or not it will actually affect those genes. So, test your understanding. Try drawing meiosis for what actually happens in the dihybrid fly with linked genes and crossing over, and show what combination of chromosomes lead to each outcome in the offspring. If you want to look at this PowerPoint separately, just look at the slides, you can go to this web address here at the bottom to find it. And it also has in that one single question to apply your knowledge. So here's a separate question about a geneticist mapping the locations of genes in plants, looking at shape, seed color, and seeing if they're linked to height. Here's the key for the genes. Here's the question and some results from an experiment to see if you can figure out which ones are on different chromosomes and which ones are on the same chromosomes. I'll see you in class soon. Happy biology!